So now we're ready for the last module of Unit 1, which is talking about the difference between observational studies and experiments. When we talk about an observational study, we've actually talked about that up to this point. An observational study seeks to observe data that already exists. We just have to figure out a way to collect it. Whether it's people's height, whether it's the length of steel rods, whether it's people's opinions about certain topics, these are all phenomena that already exist. We just have to figure out a way to collect it and observe it. When we're doing an observational study, we're not trying to manipulate our sample in any way. This would be akin to a scientist going out and if you ever see those movies, kind of hide in the bushes and just observe nature as it is. And their goal is to not interfere, not interact as much as possible, but simply to sit there as an observer. Now, of course, if we have to collect data from people, we got to interact with them. But our goal is not to manipulate them anyway. It's simply to really get at the information that's there without doing anything to the individuals within the population. One of the downsides, or I wouldn't say downside, one of the places where we can't get to in an observational is to determine exact cause and effect in an observational study. Usually that's just because there's too many other variables at play, and so it's hard to establish cause and effect simply from an observational study. Usually what we do is we collect a representative sample and then observe that sample data. Observational studies are a very poor way of gauging the effect of a treatment. If we simply look at or collect data from people who have just voluntarily taken a pill for a medical study, but there's absolutely no controls based on that, and then we just collect the data of the results, we can't really gauge whether that treatment was the cause of them getting better or not getting better. Why? Because we haven't exercised enough control over the other variables that could be hidden there in the background. Especially in the case of medical treatments, etc., we actually have to do an experiment. Now, in an experiment, we're also, of course, collecting data, but a lot of time we're actually creating new data in an experiment that didn't exactly exist before. And the purpose in an experiment is to hone in and determine if there is a cause and effect relationship between something introduced to individuals. So some vocabulary that goes along with this. The subjects within an experiment are the people or things that are being studied. Again, they're kind of like the individuals in an observational study. The treatment is what we do to those group of subjects. So it could be running some sort of visual test on them. It could be running a hearing experiment on them. It could be giving them medication that wasn't introduced before. So these are all treatments that we give to a group of subjects. A lot of times, along with that treatment, to some people within our study, we'll also give what appears to be a treatment, but really is not. We call that fake treatment, so to say, a placebo. It's a fake but benign treatment, doesn't harm them in any way, that will be given to what we call a control group. A control group is a group that we still exercise all the same controls that we're giving to the treatment group, but that does not exactly receive a treatment and instead receives a placebo. Think of it like a sugar pill given to a group rather than an actual treatment. And then factors are the explanatory variables in an experiment. Explanatory are, if you recall, are the x's in an x and y relationship. They're the ones that are explaining. And so the treatment and the factors are very similar. Treatment is just a little bit more specific, but the factors are more of a generalization of what we're after. So let's look at an example here. Many observational studies have shown, and you may have read about some of these, that people who drink a moderate amount of alcohol have less heart disease than people who drink no alcohol or who drink heavily. And in this case, we have to be specific here. A moderate amount of alcohol is being 1 to 2 drinks a day for men and 1 drink a day for women. So those people would be considered moderate drinkers. The question here is this association enough information to determine that moderate drinking is the actual cause of less heart disease? The answer is no. In this case, we did see a link between people who define themselves as being moderate drinkers and those who had less heart disease. But the drinking itself was not necessarily the factor that caused less heart disease. It just so happened that moderate drinkers tended to fit a certain demographic group that tended to maintain a healthier weight, that tended to get enough sleep, and tended to exercise regularly, as opposed to people who didn't drink any alcohol or people who drank really heavily. And so because of that, there's these variables in the background that are actually affecting the results. But all we saw, because this is all we did our observation on, was the link with the drinking and the less heart disease. So to look at a picture of this, we have our drinking habits here. These are the two things we were looking at, and the heart disease, which is our response. So this is like our X, or explanatory, and the heart disease is our response. And it appeared that we did have a link between the two. The problem is that off to the side here, we had diet and lifestyle, which was really connected to drinking, but diet and lifestyle was what we call lurking variable that was confounding our results. We say that two variables, explanatory or lurking variables, are confounded when we can't distinguish between their effects on each other. And so these two were in the background. Drinking habits was really connected to diet and lifestyle, and they both seemed to have an effect on heart disease, but really this lurking variable back here was confounding our results. So we always want to look for those lurking variables or those confounding variables in the background that might mess up our results. And this is why observational studies are not a great way to determine cause and effect. And when you listen to, especially when new sort of associations are made within the scientific community, they'll come out with these in an article or in a journal or on TV somewhere. Usually it makes it to television. And you'll hear these things being touted. But if you listen to the way that they describe these things, such as our previous example, they're very careful, most reporters are, in how they're wording it. And especially if they have guests on that are scientists, they'll always talk about there appears to be an association. They stay very far away from there appears to be a direct cause and effect relationship. A lot of times those studies have been based on observational data, not experiments. And so usually that's the first stage, is to look and find an association. And if people really want to know, hey, is, for example, drinking really the root effect? Is it the one that's having the root effect on the heart disease? They'll actually set up an experiment where we can control some of these outside variables so that we can hone in sort of on that particular explanatory variable being the cause and effect. In an experiment, if we can see that, where we're controlling all the other variables, then we might be able to say that there definitely is or appears to be a cause and effect relationship. But we cannot get that simply from observational data. So looking at some of the vocabulary we have, want you to read through this and think about each of these different things. And if we can identify what the subjects, the factors, the treatments, and the responses in the study. Here's our first one. Ability to grow in the shade may help pines found in the dry forests of Arizona resist drought. How well do these pines grow in shade? So we plant pine seedlings in a greenhouse in either full light reduced to 25% of normal by shade, or light reduced to 5% of normal. At the end of the study, we dry the young trees and we weigh them. So we've produced identical seedlings, and we've created a little laboratory where we're controlling the temperature, the amount of sun that they're getting, the soil, the geographic location, just all of those different variables that could make a difference. And we all give them the same amount of water and all that kind of stuff. So what are the subjects in this case? The subjects would be the plant pied seedlings. What are the factors? Now again, when you think of factors, think general. Then when we get to treatment, we're going to get really specific. So in this case, the factor that we're looking at, which is kind of like your variable, is the effect of light on trees and their growth. The treatment in this case, now we're looking specifically at our how we're going to run our experiment, is full light, 25% light, and 5% light. So this is the specific layout of how we're going to test the amount of light and its effect on the pine seedlings, which is our factor. And then the response, how do we measure the response of these trees? Well, we dry them and we weigh them at the end of the study. That's how we determine the effect of light on the pine seedlings. Alright, so for each of the following experiments, identify the subjects, the factors, the treatments, and the response variables again. The post-lunch dip is that's supposed to say dip, in mental alertness after a midday meal. Does an extract of the leaves of a ginkgo tree reduce this post-lunch dip? We assign a healthy people, age 18 to 40, to take either a ginkgo extract or a placebo pill. After lunch, we ask them to read 7 pages of random words and place an X over every E, and we count the number of misses. So the subjects in this case, specifically our target population, is healthy people, age 18 to 40. The factors in this case are the effects of the ginkgo plant. The treatment, well, we have the ginkgo extract, and then we have our placebo pills. So those are both parts of the overall treatment for my study. And then the response, at least what we're trying to measure here, is how mentally alert they are as they're reading. Very good. So we always want to avoid these confounding variables here. And so we're going to look at a experiment that was done in this particular school district. A college regularly offers a review course to prepare candidates for the GMAT test, which is required for most business schools. This year it offers an online version of the review course. The average GMAT score of students that took that course online is 10% higher than the long-time average for those who took the classroom course. Is this enough evidence that the online course is more effective? What type of lurking variables may exist? Think about this for a minute, pause the video, and think about what type of confounding variables we may have here and why this isn't really a conclusion that we can draw with much confidence. Well, it just so happened that this particular course one of the problems that we ran into is that the subjects were not randomly selected. In fact, this was a self-selection. the people who took the online course voluntarily took that course. And so the student population that we had taking the online course really differed from the population that had taken the classroom course. The people who took the online course that online course appealed to older working students, so people who are not just coming straight out of high school necessarily, and who also hold full-time jobs outside of class, and tended to be a more motivated group than the students' populations that ended up in the classroom courses. The other problem that we have is that we can't compare with undergraduates who previously dominated the course. Remember, this was one quarter with one class that took this, and we compared the average of that class to the long time average of those who've been taking the classroom course over the years. So you're taking one sample, which may have been 30 people, and comparing it to a sample that could have been in the thousands, right? So you have two different sample sizes that are very difficult to compare. The result of this was that the experiment was biased in favor of the online course. What we would need to do to remedy this is to exercise controls over this experiment. This basically worked out to be an uncontrolled experiment. One of the things that we would want to do is to maybe carry out this study over a year's time and then compare those results to the one-time average of the classroom courses. So they're about the same size of sample. We'd also want to randomly assign people to the different classrooms and not let people select themselves for those particular classes. And what we're trying to hone in here is to see if the treatment, and what would the treatment be in this case? Well, the treatment is the new sort of way of training people for the GMAT test. So we could see whether the online course itself, maybe the material, the training materials within that online course, is the actual cause of the increase in of 10% in the people who took the GMAT. When we talk about random comparative experiments, and it's an experiment that both, number one, is a comparison of 2 or more treatments. So we can have 3 treatments, 4 treatments, 5 treatments, etc. Where we want to randomly assign subjects into those different groups, a lot like the previous one we just looked at. The first group, the classroom group, is called the control group. This is the status quo, right? This is what's been offered up to this point. This group is receiving no treatment in a medical experiment. They're just going to receive a placebo. So in our experiment with the classrooms, the group that had been taking the regular course over the years, that's our control group, because that's our status quo, that's what's already been happening, and the variable being the new treatment method or the new teaching method, which is the online course. When we design an experiment, number one, we want to control the effects of lurking variables on the response variable. And what we want to, the way we want to do that is to make 2 comparative groups, one that's receiving no treatment or a placebo, and the other is receiving the actual treatment. We want to use random assignment for the groups, and then also third, we want to use enough subjects in each group to reduce the chance of variation. If an observed effect is so large that it would rarely occur by chance, we say it's statistically significant. Now, how we actually determine that is something that we will talk later on when we get into probability. But what we're looking for is large effects that are statistically different, enough so that it wouldn't just happen by chance. One other way that we avoid bias within our experiments from the people who are conducting the experiments is to conduct them as a single blind or double blind. We want to take the experimenters, which are usually the ones funding the research, right? They may inherently have some bias. So we want to put some controls in there to make sure that bias doesn't influence the results. If we do a single blind experiment, then the people who are participating in the study don't know whether they're a part of the treatment group or the control group. This is especially important when we're doing a medical study. There is this thing called the placebo effect, where people feel like they're getting a treatment, and so therefore they perform better, or their recovery appears to be better. They actually feel like they're physically getting different, or they're physically getting better, even though they're actually physically not getting better. But it's that sort of mental feel like I'm getting better because I'm getting some sort of treatment. By putting a double-blind level into our experiment, we're really focusing on keeping the researcher's bias out of the study. So in a double-blind, the participant and the researcher that's directly interacting with the participants are unaware of how we've divided up the groups. So if they're doing a medical examination, they don't know which group is actually receiving the placebo and which group is receiving the treatment. This makes sure that the bias stays out of both. And that's the end of the Module 4 presentation. Make sure to read also in the book, the OLI textbook on Module 4 to get some more detailed insight into some of this vocabulary. Thank you.